Hello everyone, this is Mr. Schultz and this is AP Biology video 2.4. This video covers active transport and is part of Biology in Focus 5.4 to 5.5. So, last video we talked about passive transport that does not require energy and generally goes with the flow. So it always goes with the concentration gradient. Today we're going to talk about how cells can move things against the concentration gradient. So, the need for energy. Active transport is to move solutes against this gradient. To move solutes against the gradient requires energy. And this allows the cell to maintain a balance of its concentrations. And so you might be thinking, well, wouldn't it be balanced if they just moved whichever way they wanted to balance each other out? Well, for cells, that's not necessarily the case. Sometimes they want more of one ion than another, or they want to be slightly acidic or slightly basic. So, depending on the cell and what part of the cell you're looking at, concentration gradients can be incredibly important for doing work. ATP is going to supply most of the energy for active transport. In fact, it's going to transfer a phosphate to the transport protein, releasing the energy to the protein itself. So it kind of changes the energy of the protein, which allows it to change its shape and do its work. An example of this is a sodium-potassium pump, which transfers 3 sodium out of the cell for 2 potassium inside the cell. So you end up with a net gain of -1 for your charge. And so what ends up happening is you build up a concentration gradient outside and inside of the cell so that it allows for the sodiums eventually to rush back into the cell if you open up the cell to them. So an example of this would be like in our nerve cells. So electrochemical gradients. All cells have voltages across their cell membranes. Voltage is an electrical potential energy. Cells tend to be negative by having an unequal balance. So cells tend to want to be a negative charge. So positively charged ions tend to want to move back into the cell. And the membrane potential is the voltage across the membrane. In most cells, it's between -50 millivolts to -200 millivolts, which isn't some number you need to know. Just be aware that cells tend to be a little bit negative in their charge. Both the electrical balance and the chemical balance affect the cell and its balance. And we call this the electrochemical gradient. So an example might be for concentration gradient is we have a lot of chlorines on this side, and they're all very negative. They're all a -1 charge anyway, and mostly positive potassiums on this side. So chlorines are naturally going to want to move from chamber B to chamber A, whereas potassium is going to want to move from chamber A to chamber B. However, with active transport, our cells might pump chlorine out of the cell to create a concentration gradient or an electrochemical gradient. So, an example of active transport are ion pumps. There's an electrogenic pump that transport proteins that generates voltage across the membrane. An example would be the sodium-potassium pump, where it pumps 2 potassiums in while getting rid of 3 sodiums. So 2 potassium in, 3 sodium out. And what it does is it slowly but surely builds up a charge inside and outside of the cell, where the inside is more negative than the outside is. And so this allows for ions to naturally want to move from negative to positive or positive to negative depending on their charge. Proton pumps actively transport hydrogen ions out of the cell. Think of a hydrogen ion as just being a proton in and of itself, okay? So it's the main electrogenic pump in plants, fungi, and bacteria, and it's very important in ATP synthesis. So it's actually going to be part of our electron transport chain, which we're going to learn about in the next unit. So just be aware that ion pumps come in a couple of different forms. The big 2 that you need to be aware of are sodium-potassium pumps and proton pumps, and they each work in a slightly different way. Now, there's also a thing called co-transport, and it's when a pump that transports a specific solute indirectly drives the active transport of several other solutes. An example would be proton pumps and the sucrose H+ or proton co-transporter. What it does is it pumps hydrogens out of the cell, so it creates this electrochemical gradient where there's more hydrogen outside the cell than in. So now the outside is positive, and the inside is slightly negative. So the hydrogens, they naturally want to go back into the cell, and as they do that, they kind of pull a sucrose in with the co-transporter. So what's happening is we're using ATP to pump hydrogen out, and then we're harnessing the hydrogen to pull sucrose back into the cell, which can then be converted into energy via respiration. So co-transport is an example of active transport because ATP is needed to pump the hydrogens out, and then it pulls the hydrogens back in through the co-transporter along with a sucrose molecule. Now, there's also big movement of things in and out of the cell. Exocytosis is when the cell secretes certain biological molecules by the fusion of vesicles with the plasma membrane. So here's a vesicle. It's just a pocket of something inside of the cell. And as it moves towards the outside layer, the cell membrane, it's going to fuse with the cell membrane and release whatever is inside of that vesicle. And it generally goes like this. It goes from the Golgi, which makes the vesicle with the proteins inside of it, to the cytoskeleton, which carries it to the membrane, where it fuses with the plasma membrane and releases it. This is how our body makes and releases things like insulin. It's also how neurotransmitters work, where neurotransmitters will release a large quantity of something so that it can send a signal to the next part. Endocytosis is where cells bring things into the cell. The cell takes in molecules and particulate matter by forming new vesicles from the plasma membrane. There are 3 types of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis. And it's how cells acquire cholesterol from membranes. It's also for steroid synthesis and pulling things in and out of the cell. And it's how our blood cells work. So the types of endocytosis. There's phagocytosis, where the cell engulfs a particulate or particle by wrapping pseudopodia, they're like extensions of your cell membrane, around it and becoming a food vacuole. It just surrounds it and then engulfs it. Then there's pinocytosis, where the cell gulps droplets of liquid and solutes. So it's going to pull in salts and things like that inside of a liquid. So if a cell needs a lot of something, and it's like an ion or something, it can pull a whole bunch of water with that ion inside of it to then use it. And the final is receptor-mediated endocytosis. It's a specialized pinocytosis that allows for acquiring bulk of special particulates or particles. A specific solute binds to the receptor, which forms a pit in the cell membrane and then becomes a vesicle, grabbing whatever it is that set the receptor off and allowed for the chemical to be pulled in. So just be aware that there are 3 types of endocytosis. This is the end of the active transport video, and then our final video will be about the evolution of the cell.